Hi, everyone. We're going to walk through a couple of different topics that I don't think we're going to have time to get to in class. And one of those is going to be the kidney. So I'm asking that you guys watch this. Make sure you guys do the reading for these sections as well. And at some point, we will discuss these in class as like a recap. And for this one with the kidney, we are going to do that on Thursday. So I'm going to send this out to you guys, and hopefully you watch it at some point tomorrow. And then we can go ahead and watch it on Thursday in class. I'm sorry, discuss it Thursday in class. So this whole thing's gonna be on the kidney, which is really the way that it's our body's filtration system. So the main thing that we have to be responsible for from the IV expectations is the osmoregulation or regulation of water and solute concentrations in our blood. And we also need to look at how different organisms deal with nitrogenous wastes, what those nitrogenous wastes are, and how those nitrogenous wastes actually get out of our system. So quick recap on a couple of things. Proteins get broken down into amino acids. Amino acids get built into proteins. So two different forms of metabolism. We have no ability to store excess amino acids. There's no place in our body that allows that to happen. So the kidney in all mammals is responsible for excretion or removal of those nitrogenous wastes. And so what happens to those amino acids as they get broken down, because they're a nitrogen-based compound, we have some nitrogenous waste that gets built up, and we have to get rid of that somehow. The deamination, or taking the amino group out of the blood, the amino group out of an amino acid is going to lead to the production of these nitrogenous wastes that our body is then going to be responsible for getting rid of. The thing that we need to worry about are the types of waste, who produces them in terms of which organisms produce these, and then how they are going to manage getting rid of them. Which organs in their body systems are they going to use to get rid of them? So the three types of nitrogenous waste that we're going to be looking at are ammonia, urea, and uric acid. And each of those will be described shortly. So basically we have, you got a liver here. The liver is going to help to process our broken down amino acids, remove some of the amine groups. Urea then is the waste product that we're going to look at, most specifically in terms of human osmoregulation and nitrogenous waste removal. That urea is then going to get taken out of our blood via the kidney, and then it will get transported into our bladder and then excreted. This whole process is pretty complicated, and this is one of the more difficult things that you guys will have the expectation of knowing. And so I'm hoping that this will give you a little bit of insight in terms of things that you should know and how to remember them. So one of the main things that we look at is the blood itself and what happens to the blood as it enters our kidney. And the process that we're going to focus on is something called ultrafiltration. And ultrafiltration is the process where blood itself is going to get filtered. The blood plasma itself is going to get filtered through the glomeruli. In the singular, that would be a glomerulus, and something called a fenestrated capillary. So a fenestration is simply kind of like a longitudinal flap, or an area that can kind of spread open with pressure. And so the things that are going to get pushed into our kidney are going to be things that can get filtered out by those fenestrated capillaries that happens inside the glomeruli. This whole thing here is called a Bowman's capsule. And the Bowman's capsule itself is going to contain these fenestrated capillaries, which are these teeny little blood supply mechanisms here. You have a proximal convoluted tubule, which is going to be the beginning of our kind of the internal kidney structure that we're going to look at. And then you have your afferent and efferent arterioles here. So afferent meaning blood going in, efferent meaning blood coming out. So the blood's going to go in this direction. We're going to filter some stuff out of it, and then it's going to come out and continue back to the body system through the efferent arteriole. But within here, inside the Bowman's capsule, we're going to actually have things that get pushed out of these fenestrated capillaries. Those will travel into the kidney. Some enter back into the bloodstream, but many of them stay within the kidney and then get excreted as waste. So an increase in blood pressure is going to be the main way that we can actually manipulate those fenestrations. And so we're going to have a much larger diameter of this afferent arteriole here as compared to these fenestrated capillaries. And so the fenestrated capillaries will see an increase in pressure of that blood, which causes the fenestrations to actually open up a little bit. And those openings are going to allow things to come out and then end up going into the kidney. The fenestration, we already talked about that, but the basement membrane is something that is going to prevent the large molecules, things that we don't want to get into our kidney, things like proteins. Proteins are not typically supposed to be in your urine. We're going to talk about this, but if there's protein in your urine, there's a bigger problem that needs to be addressed. But with no protein, with proteins being such large molecules, they physically can't fit through this particular gap in the fenestrations. And so the fenestrations themselves are only big enough to allow smaller molecules through. The basement membrane itself is not permeable. So this area here around the Bowman's capsule is not permeable. It doesn't allow those proteins to get out either. And so it's going to prevent the proteins from getting out into the kidney, and they're going to stay in the blood. If we look at a more zoomed in portion here, you can see the basement membrane of the capillary here kind of surrounds the whole outside. One thing I wanted you to see with this is these fenestrations of the pores. You can see relative to the size of even a single blood cell, you can see the fenestrations are actually pretty small. And so that's why we're looking at really smaller molecules themselves. That line right there under high pressure, that would actually open up. The basement membrane is kind of like the last filter as it will kind of hold things in to the capillary that are too big or not able to cross that membrane. So osmoregulation is what we call the hemostatic process of controlling water levels of all tissues in your body. So it doesn't matter what tissue we're talking about, there's some sort of osmotic control in place that's going to allow us to make sure that we have a nice water balance in terms of the solutes and the water that's in the system. Why is it necessary? Well, if we look at a hypertonic situation, if blood is in a situation where there's too many solutes on the outside, the red blood cells then are going to lose water. Water will escape the red blood cells. Those cells will then shrivel up and then be non-functional at all. If it's the opposite, where we have basically too much water or not enough solute, the low solute concentration on the outside will cause water to rush into the cells. These cells simply have a plasma membrane on them, so they don't deal with good pressure or lots of pressure very well. An increase in that pressure can actually cause these blood cells to rupture. So what we're looking for is an isotonic solution where we got equal amounts of water traveling into and out of the cells, a nice balance of solutes on the outside and the inside of the membrane. This is going to allow for the regulation of water to remain intact if it's an isotonic environment, and that is ultimately what we're striving for within our own body systems, is to have our blood itself being an isotonic solution. Consuming too much water too fast is actually something that can lead to a condition called hyponatremia. Hyponatremia, we're going to ask you to look up a little more details on later, but another word for that is water intoxication. And so literally what happens if you drink way too much water, we're talking like liters and liters within hours, not just like, a couple big glasses over the course of the day. But if you drink way too much water and you have low food intake, you're going to get way few salts in your blood as compared to the amount of water that's in there. And so we're going to get a situation where your blood will become hypotonic. Water then rushes into the blood cells, and this is going to happen all over the tissue, all tissues all over your body, including places like your brain. Whereas cells start to rupture, that can obviously lead to devastating effects. It can be deadly. This condition is something, like I said, at the end, we'll talk about things that you guys need to kind of look up. So anyway, your kidney is almost too good at filtering your blood. Much of what initially gets filtered out actually has to be returned into the blood supply. So the most noteworthy substances that need to get reabsorbed are water, salts, and glucose. If water, salts, and glucose, if too much of that gets pushed out, water and salt will have an impact on your ability to osmoregulate the glucose. If too much glucose gets out, that leads to low blood sugar, which then can cause problems, long-term effects, impact on things like cell respiration. But ultimately, low blood sugar is going to cause a problem for you because of the results of not having too much food at any given point, really. The glucose itself does get filtered through the kidney, but then the glucose itself most of that goes back in. If water isn't reabsorbed, you'd be peeing a lot more. The amount of water that's actually in your blood is really, really high. And so the amount of water that gets filtered out into the kidney, but then also gets put right back into the blood supply is a relatively high amount. Much of the reabsorption occurs via osmosis and diffusion. So simply just tracking concentration gradients. Very little of it, but some does actually travel via active transport, which is going to require some ATP. So energy is needed at some point to get water back and forth into and out of the blood supply. So this is going to be the big thing that we're working on here in terms of you understanding. This is something that is pretty complicated because what you're looking at is a single nephron, and you have millions and millions of these nephrons all throughout your entire kidney. The kidney itself is made up of 3 different distinct layers. And so on the outer layer, you can see this is a good cutaway here. So the outer layer is called the cortex, the middle layer is called the outer medulla, and then you have the inner medulla. All of these outer layers here, the cortex has nephrons basically from one end all the way to the other. You'll see at the bottom of these nephrons here, you have what is called the collecting duct. The collecting duct is going to lead into the center portion of the kidney that will then lead to your collecting ducts that empty into your bladder. So the ureters here will go into the bladder, that will store the urine, and then the urine can get expelled through your urethra. So anything where you see an orangish arrow is where things that need to cross that membrane of the nephron are going to do so via active transport. Anywhere you see a purple arrow, that's going to be places that things are going to cross via passive transport. I'm not going to click on this now because I want you guys to do this on your own at home. But if you click here, if you open up the presentation, it's saved for you in our Google Drive. If you click here, there's an awesome animation that will actually allow you to play with any one of these solutes at any single point. So you can click on all of them, you can click on one at a time, and you can see when and where it gets actually filtered out, reabsorbed, or stay in the actual nephron itself. Let's go back to this a little bit and actually look at the different locations here. And so this descending, the first thing you're going to get to right out of the Bowman's capsule is going to be the proximal convoluted tubule. That is going to lead to what we call the loop of Henle. So you have the descending loop and then the ascending loop. That is going to lead to the distal convoluted tubule. And then the distal convoluted tubule is going to lead to the collecting duct. One thing that you should pay attention to is where these things are coming out. So you can see in the outer medulla, we get a little bit of water leaving, but we get a lot of salt going across. At some point, we've got the ascending loop of Henle. You've got a large amount of salt that's coming across here. But the difference here is that, if, again, notice that active transport is what's responsible for sodium getting across in the outer medulla, but it's just naturally traveling on its own. So what would cause that? So all of these things that are traveling based on passive transport are moving back and forth simply based on concentration gradients. And so as water gets removed, you get a really concentrated solution that's inside these the tubule here. And so then as water gets removed, some of the sodium and the chloride ions and cells are going to be able to travel out. At that point, then sodium chloride is going to need some energy to get out because it's going to go against that concentration gradient. More water leaves here. And so one of the things that you'll see is that water in three different positions. So here in the outer medulla on the descending loop of Henle, here in the distal convoluted tubule, and here at the very end in the base of the collecting duct, you get water that's leaving, which is why your urine, even though it is mostly water, can ultimately get very concentrated with urea and other sort of nitrogenous waste as you are, if you're not drinking enough water. This is something that I want you guys to watch this animation and then come back with questions if you have any about what's actually happening or what's leaving and entering and why. So one of the things that we can look at is what actually is controlling that water from getting out or staying in our actual nephrons. There's a molecule, it's a hormone that we need to understand called ADH. And when you are hydrated, ADH does not get produced. So when ADH is not available, water is going to stay in the actual filtrate or in the urine. So you're going to get large volumes of very dilute urine. So you can see all these yellow dots here to represent some of the nitrogenous waste. You can see you have a large water droplet here to indicate that there's a large volume. This is going to be only the situation when you are well hydrated. However, if you are not well hydrated, if the body detects that you have a low level of water, more than it's comfortable with, or less than it's comfortable with, I should say, then more ADH is going to get produced. And when ADH is present, water is going to leave the actual collecting ducts. And so as you get to this point here, lots of water before it actually ends up in the urine will enter back into the blood supply to keep your body hydrated, which is the reason why if you are not very hydrated, you won't go to the bathroom very much. If you're working out a whole bunch and you're sweating a lot, you will not go to the bathroom very much. The urine itself will then get very low in volume and also get very, very concentrated. So you'll see that yellow or darker yellow color in the urine. If at any point you're going to the bathroom, you see that that dark yellowish color, that means that you are definitely not well hydrated. You want the urine itself to be healthier, to be considered a healthy state. You want to be at a point where it looks really dilute, meaning that it only has like a very little bit of color to it and it's completely clear. You should be able to see through it. So we're going to play a game when you guys come back, and we're going to talk about things that could end up in your urine and why that would be a good thing or a bad thing and how it got there. So I'm going to skip through this slide really quick. And so we're going to talk about protein, glucose, and urea, but we'll do that on Thursday. So diabetics obviously have one major issue, regardless of which type of diabetes is, type 1 or type 2. The issue is that they have too much glucose in their blood. So diabetics, one thing that people will start to say if you ask someone who you know is diabetic, if they were diagnosed at a later age, that they remember. They'll say that one thing they knew that something was going on is that they felt like they're going to the bathroom all the time. They were peeing a whole bunch. Because what happens is that the tissue itself gets dehydrated as you get lots and lots and lots of glucose that is in the blood. So because the glucose itself is not being absorbed into the cells, you get tissue dehydration, which leads to water in the blood. So water in the blood leads to a whole lot of water in the actual kidney, which then leads to a higher increase of urine output. But then the other thing that happens is that a lot of that glucose is supposed to get reabsorbed. It does not do a very good job of getting reabsorbed in high concentrations. And so you end up with glucose in the urine itself. So it's one thing that people will notice if they know that they were having trouble before they were diagnosed, is that they would be peeing a lot. The urine would actually smell almost a little bit sweet because it would have a lot of glucose in it. One of the things that you should understand is that is not a normal situation. Typically, 100% of the glucose should be reabsorbed in the kidney. And so if at any point there's any sort of detection of glucose in the urine, not that you'd be testing on your own just for fun, but if there was glucose in the urine, that would not be a typical situation that if you're in the hospital and getting checked up for something else and they saw that, they would definitely want to do some further investigations. There are some co-transport molecules, which would be another example of active transport for glucose that would actively push glucose against a concentration gradient. So if you see here, it says that there's too much glucose and it ends up in the urine because these glucose transporters can only work so fast. And so let's kind of walk through this and see what's going on here. So this is extracellular space. This is the actual cytosol of an individual cell. So sodium is required to actually make this kind of co-transport molecule work. And so you can see that glucose is going to be able to get pumped into the cytosol with the different sodium ions. But as you get too much glucose that's being built up inside here, the glucose itself is going to build up in the outside, in the extracellular space. You will not be able to get enough of that glucose into the cell. Too much glucose into the outside of the cell ends up in the blood. Too much glucose in the blood can ultimately end up in your urine. So insects, if you were to dissect a little honeybee like this, what would their kidney look like?
If you said you didn't know, or does it even have a kidney, then you're probably on the right track, because not all animals actually have kidneys. But the one thing that all animals do have in common is that they have to get rid of their nitrogenous waste. If you notice a little change in the background, it's because I am no longer at school. Anyway, the removal mechanism is going to be based on the ancestry of the individual organism. So throughout its evolutionary history, if its ancestors had developed a mechanism for creating nitrogenous waste and excreting those nitrogenous waste, then that organism is essentially kind of stuck on that same pathway. So you're not going to be able to take one organism that had one mechanism where it was making one type of nitrogenous waste, and then all of a sudden it's going to change even throughout its future ancestral lineage. Those future organisms will always have, as far as we know at this point, will continue to have the same mechanism of nitrogenous waste removal and nitrogenous waste production. So birds and reptiles, for example, are some that have a really unique mechanism, because if you think about the one thing they have in common, they both lay eggs, and while in that self-contained egg, that egg is not permeable. And so the things that the developing embryo is going to be producing are going to be kind of stuck inside that egg. So how do we take something like ammonia or something like uric acid and typically leave it inside that enclosed system and have it to get to the point where it's not toxic to the developing embryo? Well, you can't really do that. And so the urea that we develop, we can store in a kind of an internal storage mechanism within our bladder, and then we can excrete it. But birds and reptiles that do lay eggs, they need something different. They're not going to be able to use or produce urea because it's going to be water soluble. It's going to concentrate itself inside that egg as the embryo develops, and eventually be toxic and kill the embryo. So when we look at the things that typically get produced, ammonia is something that fish are going to produce as their nitrogenous waste. One of the benefits of ammonia is that it's very water soluble. Fish have obviously excess water available to them, and so then they can continue to flush their systems with high amounts of water to dilute that ammonia and then excrete it almost constantly. Mammals, we've already talked about urea quite a bit in terms of how mammals excrete their nitrogenous waste, but birds and reptiles are the ones that are going to develop uric acid. Uric acid actually forms as crystals. It's very insoluble, and so it's one evolutionary mechanism that's been developed to keep birds and reptiles with low water intake requirements, so they really don't need to drink very much water. And then also, while they're in the egg, still developing, uric acid crystals can, accumulate because they won't have any impact on the health of the developing embryo, because the fact that there are crystals and not soluble, it's not going to have any sort of toxic effect on the developing animal itself. That would not be true if it was producing ammonia or urea inside the egg. You'd have high concentrations of both of those substances that would eventually kill that organism. This is the same reason that kidney failure is toxic to humans. So if you can't remove the urea from your bloodstream, then the urea levels, will continue to increase, and they will eventually cause organ failure in other places, not just in the kidney. But when we take a look at something like these guys here, these little grasshoppers or other insects have their own unique little system. And so one thing that is very unusual about these insects is that they have what is called an open circulatory system. So they actually don't have an enclosed circulatory system like we do, where all of our blood and blood plasma is trapped inside veins or arteries. They essentially have organs that are kind of soaked in their own blood. So then how do they filter out the nitrogenous waste in their own systems? Well, they use these things called Malpighian tubules. And the Malpighian tubules have openings on one end, and they're essentially closed on the other end. And so as they are soaking in the blood as well, they'll absorb certain nitrogenous waste and other things like water. The stuff that is necessary for the insect is going to be able to kind of cross that membrane of the Malpighian tubule and get back into the blood. But the things that are the waste products, particularly their nitrogenous waste, are going to be dumped into their gut, and then they will excrete both feces and urine at the same time. So they have essentially a single route for both of their waste products to be removed. So that, again, is just another example of just a unique system or mechanism that animals have developed in terms of how they excrete and produce their nitrogenous waste. So when we look at the kidney and other stuff that could be in the urine, we've already talked a little bit, or at least we will when we get back together in class, talking about protein, glucose, and urea and causes for these three things. But two other things that we haven't talked about much that could end up in your blood, but typically, could end up in your blood, but then end up in your urine, which typically would not be a good thing, would be blood and drugs. Blood cells, if you go back and actually, let me see if I can scroll back really quick without making this too confusing. Let's go to this one. If you look here at the Bowman's capsule, and then we have our fenestrated capillaries in the capillary bed here, you can see that the red blood cells are significantly bigger than the fenestration. So these red blood cells should not be able to fit through those fenestrations at all. So the red blood cells will be trapped within inside these capillaries, and they'll be pushed back out into the efferent arteriole to go back into the system. So if you actually do have blood that is in your urine, it's because there's some sort of failure or breakdown somewhere else, either as a result of an infection or a tear somewhere in the kidney. Someplace blood is actually entering the kidney and it shouldn't be there. So that would be a big sign of a very large problem. Let's go back to that slide where we just left off. The other thing that could potentially end up in your urine, and the reason a lot of professional sports teams, the Olympics, as well as a lot of employers will choose to use urine testing or urinalysis as a mechanism of drug testing, is because a lot of drugs are going to be water soluble. And if they are water soluble, they'll end up in the actual urine itself. They will not get filtered out and then put back into the system. They'll actually get filtered out and removed from your system. So usually, depending on the type of drug that we're talking about, they'll be detectable within a few hours, and they could last in your system for up to a week or more, depending on the amount that would be introduced into the system and for how long it was being used. There are other drugs, for example, we talked about marijuana when we did our drugs unit a while ago. Some drugs like the active ingredient THC are actually fat soluble, and so those will stay in your system significantly longer, which is why you might have heard things like doing hair samples is one mechanism, because the fat tissue that surrounds the hair follicles will stay inside there for a long time. But drugs that are water soluble will end up in the urine, and so if there are people that are doing drugs, applying for a job, that's one thing that they would have access to, is a drug test that would be able to detect the drugs that could have been ingested in their system. So two things we didn't spend a ton of time talking about, but I want to make sure that you guys take a few minutes and check out and see what the book has to say, and then maybe do some of your own independent work on. One was that hyponatremia, which is an issue where it's another thing. It's called water intoxication, where by drinking too much water, you can actually cause damage or death. We did mention that earlier. But the second thing that I want you guys to definitely look up and at least read the section of the book, and then maybe find out some work online to see what you can determine, but is dialysis. And so if there are kidney issues or your kidneys are failing, one of the more common and more accessible things that you can do for treatment is dialysis, where they literally will filter your blood with an external machine. So I want to make sure you guys understand how that works. And then the alternative to that is a kidney transplant. So if you were able to get on a waiting list and get access to a donor kidney, or if someone is able to donate one of their own kidneys, because you can't survive with only one, you would have to be a tissue and blood match for that. Either way, you would still have to be on anti-rejection drugs for the rest of your life. But there's some info about that in the book, and I would like you to look up some additional stuff on dialysis so that we can chat about that on Thursday as well. All right. Make sure that you guys are reading and checking this out. And if you got questions, bring them with you on Thursday.